Good morning, everyone. Welcome to the Department of Biomedical Informatics and Data Science PowerTalks series. We've had lots of speakers in the past and covered different spectrum of data science and translational medicine. Today, it's my incredible honor to have Professor Jimeng Song from UIUC to give us a talk. I've known his work from just a decade ago and see his rapid ascent, developing technologies, especially related to AI, attention mechanism, and apply them to healthcare. And he's truly a leader in this field of healthcare AI, having graduated with a Ph.D. in computer science from Carnegie Mellon University. Uh, and he's currently a health innovation professor in computer science, or called a School of Computing and Data Science at UIUC, and also joined with College of Medicine, which is a very unusual setup. Previously, he was at Georgia Tech. He's incredibly well known for just his work contributing to scalable tensor analysis, graph mining, social influence analysis. Uh, but I don't think he's going to talk all about that work today. He's talking about some new topic that was really fascinated when I met him this year at the KDD Health workshop that he helped organize. So without further ado, let's welcome Professor Song. So,
Thanks. Thanks, Jake. Thanks, Amy, for inviting me to give a talk at this wonderful webinar. And so today I will be discussing how generative AI can significantly accelerate clinical trials development. We're already operating in a time where clinical research is becoming increasingly data-driven. And I believe we have the AI tools to make this process even more efficient and more impactful than ever before. So I will talk about some of the AI tools that we have developed for supporting clinical trials and to be more efficient in this talk, and show you some video demo of how those tools work. And if you have interest and use case for trying to try out some of those tools for your own work, I would love to talk to you separately and give you access to those tools. So some background before we show you the tools. And so let's first take a look at this. This plot that probably quite well known for this drug development process. We can see clinical trials are absolute essential for bringing the new drug to market. And this pipeline is very long and very complex, typically take over a decade to develop from the initial target to the final approval. And what's not super surprising, but still striking to me is how the number of candidates dramatically reduced as you progress from one stage to the next. For example, when you just started, you can have over 10,000 compounds in the screening phase. And then as you get to the phase one clinical trials, usually narrow down to just a few, like 5 candidates. And the probability of success at each stage, it's actually quite low. And for all trials, right, across all phases, and the success rate is about 50 or 60% across all phases, each phase. But if you look at only the new candidates, right, this truly new type of chemical entities, then the percentage of success is much lower, to 15% to 26%, right? So that's really highlight a key challenge, right? A single failure along this process can be catastrophic, and so can ruin the entire drug prospect. And we believe where AI can actually help in this process. There are many different opportunities I'll talk about. But if we execute this correctly, the payoff is also quite big. And for example, a single uh, blockbuster drugs like Humira can generate over $200 billion in revenue over time. And this underscores the massive opportunity and the potential impact of kind of improving this or navigating this clinical trial process more efficiently. And just a couple more background about this opportunity. If you look at the trial market, right, this illustrates a few aspects, right? The growth of the number of trials grows over time, and the size of the market and the cost. And you can see the um, from like actually the number of trials grows quite dramatically over times, right? And this is the trials in the US, and it's projected to grow to 765,000 trials by year 2027, and just in a few years. And so this almost exponential growth really underscores the increasing complexity for and the scale for drug development, right? So you really need more helps to help all those trials to be successful and globally. And second, right, if you look at the trial market, right, it's also growing quite a bit from $48 billion in 2022, and is expected to grow to 82 billion in 2027. And this represents another significant opportunity for innovation and improvement, how we can conduct those trials. And finally, if you just look at single trials, right, like single drug candidates, and breaking down the cost by phase, you can see that phase 3 is actually quite expensive, and so does every single phase almost, right? So it's, in total, it's on average, it costs close to $60 million just to go through all the 3 phases for single candidates. And so all these trends, all these trends, right, increasing number of trials and the market growth and the high cost, right, really all point to this urgent need for being more efficient, cost effective. So what approach can we bring in to clinical trial space, right, to make them more efficient? And we believe AI and machine learning can play a crucial role in this process. And before I talk about the approach, I mean, since I'm presenting in an academic medical centers, I did some research just a couple of days ago, just trying to understand where are those trials actually conducting today. And we know that most of the R&D, the money are spent on supporting top pharmas, and they're probably making most of the profit from this trials, drug development process. And we see a surprising fact, right? If you look at the number of trial sponsors and this industry, top pharma, is really a small percentage of trials, right? It's over about 3,500 trials ongoing today. And that's really attract vast majority, probably 90, I would guess 95% investment all in this space. But if you look at the number of trials are actually conducted by other, especially academic institutions, there are actually almost 10 times more. And if you're breaking down by even smaller individual entities, right, individual hospitals, you can see that, again, very interesting, surprising trend, right? That's what we see that this really a vast majority of trial running by academics institution or just regular hospitals, they don't have much resources to optimize or to kind of support even the day-to-day operation of the trials, right? This really highlight a opportunity where we see maybe AI-powered clinical trial, research assistant could be very valuable for those underserved institutions, such as academic institutions or regular hospitals. I talked to a lot of physicians in those places. Every single one of them felt that running a clinical trials is a big achievement for them, right? If some clinician can be a PI for a trial, they consider that's a kind of big deal, right? That usually can lead to some major publications, and this is very successful for their career perspective, but not vast majority of the places don't have the operation to support that clinical trial operation for their investigators. So that's what we want to change, right? To bring some AI tools to help this process. So let's really dive into what AI technology for clinical trials out there. So as you can see, clinical research is actually become very much a data science business with differences of this key stages, right? From hypothesis generation to study design to study execution to final analysis reporting. And every steps, there are some AI opportunities, right, to optimize or improve the process. And for example, for hypothesis generation, we can develop, or we already developed this literature mining assistance that can help analyze scientific literature, past clinical trial reports, to identify the promising research direction for investigator and researchers. And in the study design phase, our trial analysis system can examine the past protocols and trial-related publications and use those to optimize the trial design. Then when you execute, for study execution, we can help using AI assistance to do feasibility analysis, right? To match patient to trials automatically based on their EHR data. We can also select, if you have multiple clinical sites, you want to identify which site are optimal to run certain trials. And we have algorithms and a system for that site selection as well. And then final stage, right? Once you have the clinical trial result, and maybe along the way, you want to do some interim analysis. And we have this biomedical data science assistant and to work together with your domain experts to streamline their data analysis through code generation, like generating R code, Python code to analyze those results. And so all of this are really kind of supported by this trial foundation model that we have developed. And I will show you some of the details on that. And then I'll come back to show you some of the demo of all these different components and then answer questions. So this trial foundation model is called Panacea. So we're building this low-code small foundation model for supporting clinical trials, right? This is a joint work with my students, Jiacheng Lin, Zifeng Wang, and a collaborator from University of Washington, Sheng Wang, and his students, Han Wenxu. And so if to build this clinical trial foundation model, the biggest effort is actually collecting and processing the data, the training data. So we collected over close to 800,000 trial protocols from 14 different countries, and also gathered 1.3 million trial-related publications. That's the raw data. And then we augment those data through a kind of knowledge augmentation pipeline and to finally create over 2 million entries in a clinical trial knowledge base. And for example, some additions process we did there. We linked the trials across phases, so you know, okay, which trials, which phase 1 trial goes to the phase 2 trial, right? This linking is one part of that. We search news article related to those trials, and then run sentiment analysis about those news article to get some kind of a sentiment about the outcome of the trial, right? Whether it's positive or negative. We correlate the trial to publication, and because a lot of the results are actually in those publications, so we also get some kind of trial outcome from those publications. And finally, we also did some financial kind of linkage of those trials to the stock market changes of those corresponding sponsors, And so those are all these different sources we use to augment this data just to get the trial outcome. And the other type of data is very important is to train this AI foundation model is this instruction tuning data set. So the previous slide showed all the training data. That's to pre-train the LLM, the foundation model. And then to support different tasks, you have to create a task-specific instruction example so that you can teach the model to kind of support those tasks. For example, a task may be in this clinical trial space for matching a patient to a trial, right? You have some instruction description about the task. For example, your AI assistant, your task is to compare a given clinical notes and the inclusion exclusion criteria of the trial, then assess whether this study is relevant to patients and so on. And then we specify what type of output we want. Then the inputs include the clinical, the patient notes and the trial information, especially the inclusion exclusion criteria. Then output will be whether the patient is qualified for enroll to the trial and the explanation for that. So this is one example, right? But we need a lot of example to teach the foundation model, the LM, to understand those tasks. So here's the breakdown of those different tasks that we have created. Overall, there are over 200,000 of this type of example we curated. So that covers the study arm design. So I mean, trial search, outcome measure design, criteria design, patient trial matching, trial summarization, and so on. So this is the other data that's kind of important for building the clinical trial foundation model. And with that, then we train, here's this overview of the training process, right? We take this raw data, we did this pre-training phase, or we call trial alignment to those trial documents. And then we did this trial instruction tuning using the instruction tuning data set I showed you in the previous slide. And we can start with some of the open source based model. So we actually in this work use the Mistral 7B model as the starting point, and then go through this 2 steps to get this trial foundation model panacea. And here are some performance result on trial design and trial summarization. The first bar is the performance of our model, and the rest of those different other color bars are other open source models that we compared against.
So can I ask a quick question?
Sure, Jake, go
When
ahead.
you start with the Mistral, how much of GPU resource and the time did you have to consume?
Yeah, all this process is can be expensive. We pick a small model and then we used the like a A100, like a GPU is just a A100 one of our servers. I think we actually used 8 of those A100 and trained for about a few weeks, like I think less than months. And but that's yeah, it's
Wow.
expensive.
Okay.
But that doesn't even count the time to gather the data. Or so the data crawling processing take probably another 2 months because I mean just gather from those 14 different sites, right? To gather all those 800 trial document publication that also take a lot of time and the process.
Do you think running it once would be sufficient, or you have to actually run it multiple
Yeah,
times a few weeks?
we have to run it multiple times. In fact, some of the that's why this data processing is very critical, right? If you make some mistake there, then to correct the mistake, you have to spend the same, repeat the compute. And we did spend run it a couple of times, and it's our intention to actually continually improving this model because there are other tasks that's as we adding more and more tasks. I'll show you some of those tasks, but this is the initial set of tasks, but there are other tasks that we want to optimize. So we have to continually improving and training again. Good. Yeah, thanks for the questions. So this is kind of the high level how we train this model, and then this is kind of the extension we're building right now, right? So there are other tasks as we go and collaborate with some CROs, some pharma companies, and then realize, okay, for clinical trials, there are other tasks are kind of important. A lot of those tasks are writing support. For example, they have to in order to do the clinical trials, there's a lot of document has to be drafted to support those type of writings. For example, for protocols, for informed consent forms, for the statistic analysis planning documents, right? There's also some brochure for investigators. There's a whole bunch of document has to be produced and disseminated, right? And how do you use this type of AI assistant to draft this documents or something we're also heavily working on. And we have some initial result on that. And but yeah, this is largely kind of ongoing task. So back to Jake, your question, if you have one more new tasks, we have to improve the model again, right? Just have to fine tune those model again. And this is maybe two details, but a lot of the times you can just add additional data set to train just for that. You usually have to fine tune on all those tasks. Otherwise, they may forget about the other tasks, right? If you only train them again on just a single task. So the cost, it's not just incremental. It's usually have to be, think about it, have to do it again from a scratch almost. So that's for the AI assistant work. And I can show a quick demo for the protocol drafting work. So this is one ongoing project we have for clinical protocol drafting. In this case, we're trying to draft informed consent form. So if can you see the other screen with the video?
Yes.
Yeah. So basically, this is my collaborator and Benjamin is showing the demo. And the key part of this is actually quite straightforward. And you have a set of document, right? You have the protocol, and you have some template for informed consent that you want to kind of cover. And there's different section, what the information should go into those section, right? It's described that. And the task is actually quite clear, right? You have some protocols, you have some summary of the protocol, you have the informed consent template. Can you help draft the informed consent form? And so in this particular case, they also want this to support in multiple languages because the patients may have from a different background that may not just read English. So we have to support multi-language. But it's a process pretty similar. So we take the template and draft the automatically draft this kind of a skeleton of the informed consent, then pre-populate as much as possible the part that we're confident that we can extract from the protocol. And of course, some part need more work. And for example, you may want to explain a little bit more about the treatment, and then you can rewrite this section, right? You can say, oh, I want to rewrite this section. I want to expand this section, or you can simplify for general audience and so on. So there's some kind of rewriting capability. Then we rewrite that. If you like it, you can replace the original content with that, and then continue, right? So you can we ask this clinical research assistants or investigator to actually go through the AI generated content to verify the correctness and the appropriateness of this document. And the way, yeah, there's
another question that I have. So you build it based on the Mistral model, and then the ChatGPT o1 models keep improving, did you actually do comparisons on the writing quality, whether
Mhm.
your specialized writing is on par or better than the
Yeah, yeah. I think
constantly evolving model, or
we
yeah.
in this particular kind of solution, there are two different versions. We can use the generic state of art public model like GPT-4o or some other anthropic model, and then just do RAG, right? So they just provide this. This is perfect example for that, right? Using the protocol as data sources and just prompt the GPT to actually do the final writing. There's a version of that that's actually worked pretty well. I would say probably better than the fine-tuned small model we have done. And then so the customer or collaborator can choose which one they want to use. Some prefer local model for privacy, like a security type of thing. But if you're not have that concern of um, purring public LLM API, then this public LLM can do pretty well as well with the proper RAG prompting the correct information in there.
Oh, great.
Yep.
Thank you.
So that's a good question. And then if we finish up this demo, so the key thing is we provide references to all the information we generated. So for example, this is kind of a section for side effects, right? You might want to explain those side effects better. And then we have that expansion again. And then if you want this to autocomplete some section about a side effect, we can do that with some statistics. And actually, there's this quite detailed section that's automatically drafted, right? If you look at the evidence, right, this point to a specific table in the original protocol. So that's the evidence that the clinicians or investigator can use to verify it and confirm before they share with the patient. So that's just an example of this protocol informed consent drafting. And so I can go back to the presentation. So this is an example of how Gen AI can help you. And next, I will go to this next part about actually going to a step before the protocol drafting. This is during the design phase, right? So maybe even before you draft, if for a researcher, we actually find this literature mining quite valuable. And you have to do this literature gathering for a specific topic, understand the space, and really write a summary, right? That's can be a foundation for your research. And many people do meta-analysis, right, for in this clinical trial space, understand for a particular type of treatments, what are the evidence out there. And we have a system that focus on that. I'll show you a demo in a minute, but just go through the kind of process. You can specify what type of study you want to research, like in this case, could be a CAR T therapy for cancers, or then you can describe what you want to extract or understand what type of outcome from those trials. And then our system can suggest a set of keyword that to query the PubMed search engine. And then we also can help you to search and find those potential publications. Right? This is not that complicated, right? It's just mainly suggest a set of keywords and then use those keywords to construct the right API call to PubMed API to get the publications. The next step is more actually we found more valuable and important. Then you have to read those papers, right? That's where a vast majority of times people have to spend, right, to read those individual paper to extract information and also confirm whether those papers are truly relevant to your study. So we do screening, right? You can specify the screening criteria, right? Okay, you want to find what type of study, what type of outcome measure, and so on. For example, I just want to focus on phase 2, phase 3 trials, right? That could be a screening criteria here. And then we can score using AI assistant to read those papers for you and score them against all those different criteria. Green color means they passed the criteria. Red doesn't, it means they failed. Gray means we don't find evidence in that paper to support either way. And then you can probably select the ones that have most of the passing and use that as a starting point for detailed analysis. The final step is the detailed analysis, where we can still help you a little bit, right? Where you can read those papers at that point, right? That may be like 10, 20 papers at that point. But we can also extract some important measure you cares about, such as this complete response rate, partial response rate, and so on. So this is the most difficult part of this pipeline, because to calculate those measure, we don't just retrieve that, right? Because a lot of paper don't say that explicitly, so we have to calculate that with programs. So that's where the difficulty comes in. Then at the end, you'll get all this result, and that's how it works. And I think I can just give you a little bit more details about the pipeline, where we take all this publication data, and our could use our model or GPT or some kind of public available model for this to do with RAG. And the goal is to, okay, in this particular case, we actually have curated another benchmark data for optimize this task. So we take 21,000 meta-analysis or review publications, right? And involve some for some clinical trials. Then overall, there's close to 405,000 publication individual studies there. And then we want to kind of reproduce those review papers, right? The findings in those papers, that then validate our automatic pipeline for doing that. And there's all those steps I mentioned earlier. This is just one part of the most challenging part for the result extraction, where, for example, here, this is the things that we extracted from the original paper, right? This says all 6/9 patients showed objective clinical response, 2/9 achieved complete response, right? This could be the result from the paper, but to turn that into a number, or we have to do small calculations and to know, okay, this response rate is 0.22, right, in this case, so that you can plot that in a forest plot, which is quite common in a lot of this review, a systematic review or meta-analysis paper. And here's some result in terms of the kind of performance for search, like finding all the relevant papers were a lot higher than if you just give humans some fixed amount of time to do Google search, or just even worse, right? If you ask GPT, today GPT can do web search, but it's not optimized for recall, right? It's optimized for maybe precision, right? Maybe even that's arguably maybe not true, but it will give you a few paper. If you want to find 50 papers, I don't think you won't get that result, right? So that's why GPT performance is very low. And human need take a lot more time to gather those paper. If for just a short amount of time, they won't be able to do that either. But use our system, we can get close, I mean, over 90% recall compared to the original review article, right? If the review article have a 50 paper or 100 paper, we can cover 91 of those. And here's some result in terms of this is human preference analysis, right? I just show them example of output of our result and ask user to verify which one they prefer. Vast majority, they prefer our output. So, okay, so is there questions or? If not, I will just continue. And I think maybe I think I explained that a little bit. I think if I've, you see, maybe in the interest of time, I won't show the demo because demo pretty much takes another 7 minutes. Just go through the pipeline I explained. Happy to share the video so you can see how it works, but I will show you a few other example. And this next one is a, for trial design. For a specific part of trial design, that's eligibility criteria design, then using those criteria to do a feasibility analysis to find out how many patients actually in your database that satisfy those criteria, right? So that's where we help the process is quite similar. You have some criteria, sorry, some trial information that's you're thinking about doing, and then we use that to query similar trials, that two-year trials, and explain why they are similar to you, and then extract the eligibility criteria from those similar trials, right? Then organize those criteria into different groups based on some kind of a clinical similarity of those criteria. Maybe one cluster could be a disease condition, right? Another maybe demographics, and so on. And then you can pick and choose from this set of clusters of criteria from similar trials and then add it to your new trials. Or you can use our automatically recommendation algorithm to suggest a bunch of criteria based on what's in those similar trials. And once you have that, that will be the kind of the criteria you drafted. And then you may want to test it. Okay, are there patients, right? If you use those criteria, are there enough patient left, right? So that's involved. This part is actually still ongoing. It's quite tricky and for various reasons. Happy to drill down. But essentially, we have to write code, right? Take each criteria, then convert that into a program, then query your database to see how many patients that satisfy those criteria. So we do this 4 steps, and eventually you will know, okay, what criteria you should use for your new trials and what's their impact, right? To if you have a patient database and how many patients will still satisfied with all those criteria.
So I have a question about the criteria picked. So I was thinking about the Amazon shopping situation. The criteria usually are based on some kind of ontology,
Mhm.
and then these criteria were searched among the maybe the previous trials. Some of them may be obsolete, some of them may be late breaking and more applicable. How do
Mhm.
you like find out which one should really be like suitable, or maybe it's very much of a dependent on the data. To some trial may pick those criteria because they don't want too many patients. Some maybe just a so small disease and they don't want to include those.
Right, right. I think this process is very today is very ad hoc, right? So most of the designer have their own preferences or biases, right? They felt that can increase the chance to make the trial more successful. So they usually balance this to be very, very specific. So the cohort can be quite small, but they felt, okay, maybe this my drug is will be optimally be successful for that cohort. But then the other side will be you have to balance the availability of or feasibility of that cohort. Right? If there, you don't have enough patient, then the recruitment will be very difficult, and also there's a meaning that will actually take longer if you only don't get to the right enough samples, right? You may still fail just because sample size like statistically not enough to power the result. So there's all this trade-off. Today is really ad hoc. I mean, we talk to a lot of people are just very ad hoc. People have different preferences. And what we did is very at the very high level, that's what they did, right? Look at similar study and what they use, then try to create. Then at that point, they will introduce some of their intuition, or maybe do some secondary analysis, right? Look at maybe the preclinical study or the previous phase study, right, to get some hints. But this is, I agree, still very ad hoc. At least we want to make this steps are pretty standard. So make this a few steps efficient, so that at least they can find similar trials very quickly, narrow down to the set of criteria. Then they have to decide which one to use. And we can, if the last step, if they have the patient database and we can do this, then they have at least some sense that whether those criteria will be too strict or not. But I agree with you, there's still a lot of nuance, right? How do you pick from this list, right? There's a lot of justification probably they have to manually investigate. And so that's this few steps, right? In terms of the backend research, what we have done is we have this trial search capability, which is very essential for all this Gen AI RAG-based approach, because you always have to find similar trials. So there are some details about how do we decide which trials is similar to your trials, right? There's trials document is very long, and if you don't pay attention, and the different sections have different importance, right? And so you really have to figure out a way to provide the right weight for those different sections. For example, the intervention and the condition may be much more important than some other background sections, right? So we have this kind of hierarchical encoding to help to do that, and to have this encoder, right, taking care of all these different sections. And then label training data is very limited. So we have to be creative to create the supervision. In this case, we use some self-supervised learning approach to create those similar trials, pairs, and the dissimilar pairs. And so that's kind of the work you have to do. So there's some results showing we compared to a lot of bird-based, other type of embedding-based method, our approach is much better in terms of accuracy. And we also organized all those trials in a natural disease cluster if you plot them. So that's another kind of validation on that. So the last part I really want to talk about is this trial matching. This is a very common task almost for every trial, right? You have to find the patient. This is always a tricky, time-consuming process. And this is collaboration with NIH, with researchers there, Qiao Jing and Zhiyong Lu, and their clinicians, Dr. Faladas, and also with my students, Zifeng Wang, for this work. And this work, I think, just got accepted to Nature Communication. So basically, the idea is, can we use LLM to help to do the matching between using the patient electronic health record data, especially the clinical notes, and the trial criteria. So there's two different use cases of this. And if you have starting with trial, you want to find patients, right? That's a lot of this pharma trials, right? In this settings, you already have the trial, it's already designed. You want to find the patients. And the other view is also becoming increasingly valuable, right? Patient may be looking for trials they are qualified. So there's just a single patient's record we want to match against a set of relevant trials. And for a lot of hospital, academic institutions, we talk to clinicians, they want a second option for their patients, so that their patient can see what are the most relevant trial to them very quickly. And that's this work is about. So we take the patient notes, and the process is actually quite simple. I mean, and you have this patient notes. We break them into sentences, and then we take all the criteria into two set of criterias, inclusion and exclusion criterias. Then we do this two-step matching. And step one is just criteria level, individual criteria level matching to patient notes. And then we produce this output, structured output, to highlight just criteria by criteria, this patient match to that inclusion criteria or exclusion criteria, and which sentence is the evidence for that. And so with that, we will do a second level process to aggregate all those matching results to rank them. Basically, now you can give some kind of a score for this entire patient to this trial, so that can be used to kind of rank different trials for this patient. I think the latest result is probably better, as we iterated that paper a few times already over the review process. But now, initially they have this about 75% accuracy for this matching part. Then for exclusion part, there's about close to 70% accuracy. So that's the trial GPT work. So that's pretty much the final thing. And then there's one more thing on the data science assistant. The paper is still being developed, but I think the tool is very cool. So I will just show you the tool, and then I'll just conclude. And so we have a lot of collaborators are biomedical background, so they don't necessarily have a very strong coding background, like programming background, but they have data. They want to analyze the data, right? So if, for example, you have some TCGA data, you want to do some kind of analysis, and that's something we want to support, right? You upload the data, you can browse the data, and there's a few tables there, and then you can ask questions. I'm just fast forward to the coding assistant part. There's different type of assistant. Let's say you want to visualize the distribution of expressed genes, mutation, patient overall survival time in the data set, and show me the size of data set. Or you can just ask natural language question, and this will lead to some programs. Or so we generate some Python code, and it can be quite long because you ask a bunch of questions. And then we have a sandbox. You can run it, and then so just on their environment, and you don't have to set up on your local environment or copy the code to your local. So you can actually run it there and see if it works. And then it would generate some of the result, right? This overall survival time and other analysis there. So it's actually could be quite useful tools for analysis for data scientists. And I'm just fast forward for another thing. This is maybe you want to plot oncoprint, right? This is some genetic data. So this is a very specialized analysis. We still can do that because we have trained our model with a lot of the biomedical code base on GitHub. So this is in those examples, right? So you can generate the oncoprint for that and so on. So this assistant can also link to other knowledge base. For example, we're just querying NCCN guideline, right? You want to understand what's the first line treatment for breast cancer, right? They will read that guideline and give you some answers there and provide the references for that. So just an example of how data scientists can work today, right? Looking at, they do some coding, data analysis with data, then they do some research on publication or clinical guidelines, right? Back and forth, right? To do this. So our software can support both. I think that's it. Just to conclude and have we have developed a suite of AI tools addressing various aspects of clinical trials need, from protocol drafting to patient recruitment to evidence synthesis to data, coding. And these tools have potential, to significantly accelerate clinical trials, improving the efficiency of the trial process. And my hypothesis is this tool could be particularly valuable for academic institutions and other under-resourced organizations. So with that, thank you for having me here. I'm happy to answer questions.
Yeah. Well, I just want to make a comment. This is really amazing, amazing journey. So I know that there's probably a company behind it, but I rarely see that the technology is built on solid research and every part is really just great research, and then you have software product that can be used to do useful things. So I wonder if you can tell us which of the 5 ones are readily available for academic use and which one we need like commercial licensing and partnership to do.
Well, I felt we developed this. I think all of this we can, on a one-on-one basis, we can offer this to our academic collaborators mostly. For free. I don't think we want to make money from. We probably can learn a lot from your use case as well. In fact, we work with some of our MD students, our faculty at the UIUC on some of this, but we don't intend to really kind of make money from them. But at least not right away. And so we have some customer from more bigger pharma and CRO, so they are actually have bigger contract we work with and to support some of the development of this. But I felt this technology is valuable and for individuals who are a researcher and academic. So if
Great.
you're
Yeah.
interested,
Yeah,
do
we
come.
will be in touch. And then we're two-person raised the hand. Who first and then Ryan?
Okay,
Go
Rahul.
ahead, Rahul.
Uh, hi, Dr. Sun. How are you? It's really nice to see your talk. I have been working on this clinical trial digital twin projects for. I was a postdoc with Dr. Chen. I've been working on this for long, and I'm really inspired by your work. I have been doing some work for biomedical analysis using large language model, like talking to the databases, getting information in chunks. And I can see that there is a lot of stuff that you have done. And I have so many questions. The only question that I would like to know, from where you get all that data? Is it available, like the clinical trial data from NIH and other sources? Because I am thinking in the same line, but targeted to some specific things like selecting patients. So I want to target on that part because it can be used in two ways. It can be used for clinical trials as well as it can be used for precision medicine.
Yeah,
so
yeah.
patient selection, and I have a targeted work focused on that. And I want to know that how do you get that kind of data?
Yeah, the data is all public available. You have to crawl some of those data, but we actually curated some benchmark data set you can download directly. Those are processed the trial data. But we're happy to talk to you if you need help with some other type of.
I will already send you a LinkedIn request, and I
Uh-huh.
would like to connect with you on the sidelines and know more about this because I'm really into this research.
Okay, great. Thanks. Thanks.
Thank you so much.
Ryan. Yeah.
Yeah, thank you for this. This was fantastic. I'm super excited. You may have answered my question actually with your QR code. So I'm going to scan it right now if you'll go back.
Okay.
Actually,
but I was curious.
could you share your slides? I think a lot of people are interested in your work.
Sure, sure. I will send a PDF of this slide to you and
Great.
after the talk.
Yeah, that's fantastic. I was really wanting to follow up on Jake's question. Which of your tools do you have like available? We could go play with them right now. Which ones do we need to like request from you? Which ones would we need to become collaborators with you? Like, yeah.
I think it's mainly the resources we have to support that. In terms of the maturity of all these tools, I think this data science one I showed the last is probably the most mature, and also it probably cover most of those like capability. I mean, that's in the other steps as well. Like it has the trial search, summarization stuff, publication search, coding assistant. It's just more complete anyway. So we're focusing a lot to get this out because it's the most generic version, broader version of this. So I think it's some version of this. It's can be, I mean, provided if you contact us, and but it's still heavily under development. So that's why we didn't put it out for the public because just support. It's a
No, that makes sense. I'm asking specifically because I know there are at least three separate groups at UAB, each independently working on a clinical trial screener using AI.
Mhm.
And it seems like you've already done like most of the thing they want to do.
Yeah, I'd love to collaborate on that, right? So we had this, I mentioned this trial GPT work with NIH that's coming out from Nature Communication. That work is definitely ongoing. One big challenge right now is that thing is costly. If you want to screen 1 million patients, the current framework is too costly. So we have to do something else on top of this to make it more efficient. So that's something we're working on actively, but happy to collaborate, right? Just on that topics alone.
Okay, awesome. Yeah, I think your contact information is somewhere. We'll probably reach out. Thank you.
Yeah, this is my email for everyone on
Perfect.
call, right? Just Jimeng, just my first name at illinois.edu. Feel free to email me. Happy to collaborate.
Yeah. Well, I also want to mention that at UAB, we have this new center, Systems Pharmacology AI Research Center, and your work is very much of in line with and complementary to what we do. We're more on the upstream disease biology mechanism finding part, but then the clinical trial is very much complementary. So if you're interested, I will reach out to you and see how we can work together as a
Oh yeah,
collab.
I'd love to. Actually, I think Jake, we talked about this. I want to get connected to you guys quickly. So we did this online seminar. I'm happy to visit in person. In fact, I come to because of collaboration. Other reason, I come to Atlanta very often. So I'm happy to come to UAB. I
Yeah.
mean, in
Okay.
future.
That's fantastic. Let's keep in touch offline. I think this is really amazing. You're setting a trend how the healthcare AI is happening. And for those of you who don't know, he's one of the top 100 person in healthcare AI researcher. So I shared your talk to ISMB years ago and following your work, and this is really amazing.
Thank you.
With that, I thank Professor Sun again. Okay.
Thank you. Thank you, everyone. Thank
Okay,
you,
great.
Jake and Amy.
Okay. Bye.
Bye bye.